Hello everyone. Today I am going to talk about one of the important techniques of molecular biology, that is PCR. What does PCR stands for? P stands for polymerase, C stands for chain, and R stands for reaction. Polymerase, chain, reaction. So this PCR is an automated machine. Where the DNA synthesis will take place under in vitro conditions. You all know the difference between in vitro and in vivo, right? Here we are saying that the synthesis of DNA takes place in vitro conditions. Okay, in vivo means what? That is within the living organism, but in vitro means that is within the test tube. So here in this instrument we are going to set a different temperatures, means the different steps are going to take place that finally results in the synthesis of a DNA. So in this method of PCR, so we are going to set up a reaction in a test tube that is going to be placed into this machine, which we call it as a PCR machine. And then with a series of steps, finally what we are going to get at the end, we are going to get millions and billions copies of the DNA. So simply we define PCR as the polymerase chain reaction which is used for the amplification of DNA. Okay, we are saying that the amplification of DNA, nothing but making the multiple copies of DNA. For example, you have taken a DNA and this DNA will be used as a template. So when you incorporate this template DNA along with the other requirements to start this procedure, at the end what you are going to expect, you are going to get millions and billions copies of the DNA at the end of the 30 cycles. We are going to set alternate temperatures from high temperature to the low temperature, and then we are going to carry out the synthesis of DNA in this thermocycler. So this PCR can also be called as a thermocycler. Hope you understand what I am saying. This PCR is an instrument which is an automated instrument where the synthesis of DNA can be carried out with more accuracy. This is more rapid and an accurate method for the synthesis of DNA. So for the better understanding of this topic, I am going to provide you an iCard. Please go through my video on DNA replication. Earlier in my previous video we have seen about the DNA replication in bacteria, that is in vivo conditions. That means that process of replication which takes place within the organism. Here we are talking about the DNA synthesis in vitro conditions, that is we are going to set up a reaction in the tube and that we are going to keep it in this thermocycler which is maintained at a different temperature so that we can expect billion copies of DNA at the end of the PCR. Okay, if you have incorporated a single DNA and after one cycle you are going to get 2 copies of the DNA, and after the next cycle you are going to get 4 copies of the DNA, and then 8, 16, 32 and so on. Okay, every time if you observe, the concentration of the DNA gets duplicated here. So the strand here which we are providing, that DNA will act as a template for the synthesis of new strands of DNA, and in the next round those newly synthesized DNA will act as a template for the synthesis of again the new DNA strands. In this way the process will continue till 30 cycles. So what can we expect at the end of 30 cycles? We can expect billion copies of DNA at the end of 30 cycles, and that we are calling it as amplification of DNA, which is achieved with the help of an automated machine called PCR, and we can also call it as a thermocycler. Okay, and by this technique rapid cloning can be achieved. As I told you, by using this technique we can amplify specific DNA. From where can we get that DNA? Okay, so nowadays we can easily get the gene sequence from the libraries as well, and by using this technique rapid cloning is possible. What is meant by cloning? We are saying that rapid cloning is made possible with this technique. So cloning is also nothing but making a multiple copies. Here in this method of gene cloning, what exactly happens is, example, this is the DNA, okay, and here we are having gene A, gene B, gene C and gene T. Okay, we are having 4 genes here for example, and you are interested in gene C and you want to make a multiple copies of this gene C by utilizing a method called gene cloning. So what you are going to do it here is, first you need to isolate the gene. How will you isolate the gene? The thing is like you need to use some enzymes called restriction endonucleases. What the nucleases will do? They will degrade the DNA, right? So we are not interested in gene A, B or D, but we are especially interested in this gene C. So we want to isolate this gene and go for gene cloning, so that we want to make a multiple copies of this gene C. For that we are going to treat it with the restriction endonucleases. Okay, so what happens then? We'll get a desired gene of interest. So this is our gene of interest, gene C. So next step is, we need to introduce this gene into a plasmid. Okay, this is a plasmid which is present in a bacterial cell. Okay, this plasmid have a replication property, right? When it undergoes the replication, the gene which we incorporated will also get replicated, and at the end what happens? We'll get more number of copies when this plasmid undergo a replication. And for this purpose again we need an enzyme called ligase. Okay, listen carefully. Here we are isolating the gene by treating with the restriction endonucleases. Then we are having a gene of interest and we have a plasmid which will be used as a vector here. So we need to incorporate this gene into a plasmid to make a recombinant molecule. So whenever this plasmid undergo replication, our gene will also participate in the replication along with the plasmid. So here, as a process of gene cloning, we are going to get the multiple copies of this gene after the process of replication. And if we want to join this gene with the plasmid, again we need to use ligase enzyme. Okay, so this, remember, this gene cloning can occur both in vitro and in vivo. Okay, we can carry out this process within the living organism as well. So we can perform this experiment both in vitro and in vivo conditions. Okay, and here if you observe, for PCR we don't need any restriction endonucleases and we don't need any ligase enzymes, or we don't need any vector. And here what happens? So after you introduce the gene of interest into the plasmid, next step is you need to introduce into host. Say for example you use E. coli. So once the plasmid is made recombinant molecule, like after the insertion of the gene of interest into the plasmid and you generate a recombinant molecule, what you need to do next? You need to introduce into a host, and then when it undergoes the replication, you are going to get a multiple copies of the gene. Why you need to get a multiple copy of a gene? For example, I already told you are interested in gene C, right? It may code for a useful protein. Say for example, it code for some immunoglobulins, or it may code for a hormone insulin which you are interested in to produce in a huge amount. But earlier with this single gene you can't produce sufficient amount of insulin. If you want to make high amount of insulin, you need to go for a method called gene cloning, because here you are going to produce a multiple copies of the gene. From those multiple genes you are going to extract the insulin hormone in sufficient amount. So this is all about the gene cloning. Okay, now you are clear with the DNA replication that takes place under in vivo conditions, that is within the organism, and now you are clear with the gene cloning method. Here now when we come back to the topic that is about the PCR, we are saying that this technique makes the rapid cloning possible. Why? Because this process, that is the synthesis of DNA, takes place in vitro conditions, that is in the test tube. Once we add all the reagents to the test tube, providing the DNA as a template. Okay, so this is an automated instrument where we need to set up the reaction, and then the DNA synthesis will take place, which finally at the end of 30 cycles we get billion copies of the DNA, right? And this PCR doesn't require any restriction endonucleases, ligases or vector, anything, as all these are the expensive method, and moreover it is a time taking process which may take around 2 to 3 days, but when it comes to the PCR it can be completed within 15 minutes, right? So that is the beauty of this technique. So simply we say that the PCR is an automated machine which carries the synthesis of DNA in vitro conditions, especially for the amplification of the DNA, that is to make a billion copies of DNA, right? This PCR was invented by Kary Mullis in the year 1985, and he was awarded with the Nobel Prize. I personally feel that when we talk about any technique, it is very important to discuss about the scientist who is behind that technique. It is their efforts. Today we are depending on the technique. I'll tell you a short story about him. Okay, if anyone knows about his story, you can just skip the video, right? This person, Kary Mullis, has graduated from the University of California, and initially he was not much interested in science. His interest was in music, dancing and in sociology, and moreover initially he worked on iron metabolism. Actually his work is not on DNA synthesis. Initially his work is on iron metabolism in bacteria, that is especially he studied on siderophores, right? And then at the time of submission of thesis he felt some difficulty because he doesn't have much knowledge in the field of molecular biology. Later, to prove that he is interested in science, he has written a manuscript, and that was published in a journal called Nature, which is a reputed journal. So with his publication he successfully defended his thesis, and he got awarded with the doctorate in the field of biochemistry. After that he did job for quite some time. Later again he left the job and he somehow managed to run a bakery for 2 years. Again after 2 years of time period he came back to the field of science, where he worked with Dr. Joe, and there actually he started working on DNA synthesis, right? Earlier he doesn't have much knowledge on molecular biology. Later he started working on DNA synthesis. Then one day what happened is he went on a drive with his girlfriend. While he was driving, he has lot of ideas in his mind. He started thinking that instead of one oligonucleotide, why can't we add 2 oligonucleotides into a reaction mixture? In that case, one oligonucleotide will go and bind with the upper strand, and one oligonucleotide will come and bind with the lower strand. And in this way, if a DNA synthesis takes place, how it would be? So that is the kind of thinking he had at that time. Suddenly he stopped his car, took a piece of paper and pen, and he started calculations. And he calculated till 30 cycles. And he has this idea of DNA synthesis utilizing a technique of PCR. right? But he was not sure whether this idea would work out or not. He went to a library, he searched for so many books to get a literature on the amplification of DNA, but there is no literature supporting this data. Then two lab technicians who is working with him has come forward to help him. So these people worked on this concept for a period of 3 months. So after 3 months, kary mullis could set up this PCR reaction for the first time, and he gave all these findings to a journal called Nature, but they refused this data. He felt very upset. Later his findings were published in Methods in Enzymology. That's the story about the kary mullis. So it's his thought, it's his efforts which we are making use of them right now. Thankful to him, and let's move on to the topic back. Okay, so I am saying that this PCR is an automated machine which carry out the amplification of DNA under in vitro conditions by a repeated cycles of strand separation and the synthesis of DNA. So what happens here? It is set in such a way that we are going to carry out the repeated cycles of strand separation and the synthesis of DNA by providing certain reagents. Like we need to provide a DNA template, we need to provide an enzyme, that is DNA polymerase enzyme, and we need to provide DNTPs and also primers, forward primer and the reverse primer. So we are going to provide 2 set of primers. I'll explain you each in detail. The 3 important steps that are involved in a PCR method are, so the first step is denaturation, we can also call it as strand separation, and the next step is hybridization of primer, or we call it as annealing, and the third step is extension and DNA synthesis. So this is the final step of a PCR where the actual synthesis of DNA takes place. So let's see each step in detail. So the first step to carry out a PCR is denaturation. What do you mean by denaturation? See, this is your double stranded DNA. Okay, you'll have bases here A, T, G, C, T, C, G, A, G, C, like this for example. And what will be the sequence in the opposite strand? As you all know, both the strands will be complementary to each other, and adenine always pairs with thymine and guanine always pairs with cytosine, right? See, in between adenine and thymine you'll have 2 hydrogen bonds, and in between guanine and cytosine you'll have 3 hydrogen bonds, right? So this is a double stranded DNA. Double stranded DNA which you are going to use it as a template, and you need to provide this gene in your reaction mixture. Okay, so the information that is present in the DNA will be utilized by the DNA polymerase, and it is going to add one nucleotide after the other, and it is going to synthesize the new DNA. So the action of DNA polymerase is what? It is going to catalyze the synthesis of DNA, right? So in a reaction mixture, what all you are going to add? So what all you are going to add? DNTPs, you need DNA template, you need set of primers, you need Taq DNA polymerase, of course you need water when you set up a reaction, and along with this you need to add some tris HCl buffer to maintain the pH, and sometimes you can also incorporate magnesium and potassium, which will be used as a cofactors for the enzymatic activity. So first thing is you need to set up a reaction. Just listen, I will tell you the procedure in detail again. So once you are done with all these requirements in a test tube, what you are going to do, you are going to set up a reaction with the first step of denaturation. So I'll tell you first the 3 steps, then when we talk about the procedure I'll explain you again. So here the first step is denaturation or strand separation. Here what going to happen? So when you apply a temperature of 95 degrees centigrade for 15 seconds, what going to happen? The strand is going to separate. This is a double stranded DNA, right? Both the strands are going to separate. Once you apply heat, that is you are going to maintain a temperature of 95 degrees centigrade for 15 seconds. Okay, so what is going to achieve when you apply this temperature? You are going to separate both the strands. Okay, this is how both the strands will separate when you heat the solution to 95 degrees centigrade for 15 seconds. Remember, in some papers you may see a temperature of 98 degrees centigrade for 1 to 2 minutes. So in some other protocols you may see a temperature of 95 degrees centigrade for 15 to 30 seconds. So this time and temperature may vary depending upon the nature of DNA and the salt concentration that is present in the buffer. Do you understand? Some papers has mentioned the temperature of 98 degrees centigrade for 1 to 2 minutes and some papers has mentioned the temperature of 95 degrees centigrade for 15 seconds. Here what you need to do is you need to standardize the temperature, because the temperature and time may vary with the reaction, vary with the sample of DNA. For example, if you take a mammalian genome, here you need to apply a higher temperature of 98 degrees centigrade, and if the DNA has more amount of G plus C content, that is around greater than 65% of G plus C content, again you need to apply a higher temperature of 98 degrees centigrade. And sometimes generally the salts are required for the activity of DNA polymerase, which is responsible for carrying out the DNA synthesis. And even with the more presence of salts also, sometimes we need to set a temperature of 98 degrees centigrade. So usually the temperature ranges from 95 to 98 degrees centigrade. Why we are maintaining that temperature is to break the bonds, that is we are going to separate both these strands. So each strand will act as a template for the synthesis of new DNA, right? So what will be achieved in the first step? In the step of denaturation, we are going to apply a temperature of 95 degrees centigrade for 15 seconds, and at the end what you are going to get? You are going to get 2 separate strands. And here remember one more important thing, that is we are applying a temperature of 95 degrees centigrade, right? Generally the DNA polymerases are very sensitive at that temperature. So for that reason only, especially for PCR method, we are going to use an enzyme called taq DNA polymerase. Okay, this enzyme can resist the heat, right? So but actually the half-life of this enzyme is only 30 minutes at this 95 degrees centigrade temperature. So you make sure that when you set a temperature for denaturation, it shouldn't be more. Just plan an experiment that it should last for few minutes only. That is, you can set it for around 15 to 30 seconds for the denaturation of the DNA, where you can separate both the strands of the DNA, which will be used as a template for the synthesis of new DNA. Okay, and coming to the second step of PCR, that is hybridization of primer, or we can also call it as annealing. Remember, primers are the short nucleotides with a 20 to 25 nucleotides, and these are prepared chemically. How will you prepare a primer? There are many softwares available to design a primer, and for designing a primer you need to know the Tm of a primer. Tm stands for melting temperature. So to define a melting temperature of a primer, it is a temperature where 50% of the primer and its complementary sequence should form a duplex. I'll explain you here. We are saying the hybridization with the primer, right? So after the separation of the strands, that is in the first step what we achieved, we got two separate strands because the duplex is converted into a single strand by applying a heat of 95 degrees centigrade. So this each strand will act as a template, right? To start the process of synthesis, what do we require? We require RNA primer, right? And these primers are synthesized chemically having a knowledge of the melting temperature and as well as the sequence information that is present in the DNA. Okay, especially if you want to amplify a specific gene. For example, this is A, B, C, D and E. Some 5 genes were there on the DNA, okay, and you are interested in amplifying this gene C, and what you need to know is you need to know the sequence of these flanking regions, okay, forward primer, and we need to design a reverse primer, okay, and we need to know the sequence that is present in the flanking regions of this target gene. Then only we can design a primer, so that the primer will go and bind over here because the sequence is complementary to it. Okay, for example, if the sequence here is ATGC, we need to design a primer with the sequence of TACG. That means both these sequences are complementary to each other because A always pairs with T always pairs with A, guanine pairs with cytosine, and the cytosine pairs with the guanine. So with this information we can design a primer so that it will go and base pairs with the complementary sequence. Okay, this is nothing but the hybridization of a primer. Hope you understand. And remember, the annealing temperature is maintained at 54 degrees centigrade for 30 seconds. So from 96 to 98 degrees centigrade, we are going to reduce the temperature to 54 degrees centigrade. Okay. So the reason is here at this temperature only the primer will go and hybridize with the complementary sequence that is present on the DNA. That we are calling it as hybridization of primer. And for designing a primer, you need to know the Tm value. That is the temperature at which the 50% of the primer and its complementary sequence will form a duplex. Okay, because the strand is now a single strand. So this primer should go and bind with the DNA forming a duplex so that it will initiate the synthesis of the DNA. We all know that for a replication process we need to have a primer to initiate the process. So here we are going to provide the primers, and those primers are to be designed by us with the information that is present on the template DNA. If we are especially interested in a certain gene, we need to know the sequence of the flanking regions that are present adjacent to the gene of interest. Okay. Some people said that the Tm value can be calculated with a simple equation of 4 into G plus T plus 2 into A plus T. So generally this formula can be used to calculate the melting temperature of a primer when we design a primer. But I don't think this is an appropriate formula. It doesn't work well with some researchers. And there are many other formulas where we can use them to calculate the melting temperature of a primer. Even many softwares were there where we can calculate the Tm value. And always remember that when you want to standardize the experiment and when you want to standardize the temperature of this annealing procedure, you need to know the Tm value, and based on that Tm value you need to use a temperature here approximately less than 5 degrees centigrade of Tm value of a primer. Hope you understand. First thing is you need to check the Tm value, and then you need to set a temperature here for annealing, and that should be approximately less than 5 degrees centigrade of the actual Tm value. And this is how you are going to initiate the hybridization of primer with the template DNA. Okay. Coming back to the third step of PCR, that is extension, DNA synthesis. So here the actual synthesis of DNA takes place. What happens in the first step? We are going to separate both the strands of the DNA by applying a heat. In the second step, what happens? The primers which we provide in the reaction mixture will go and bind with the single stranded DNA because both are complementary to each other. And what happens in the third step? The primer will initiate the process. Tag DNA polymerase. So we discussed about DNA polymerase 1, 2, 3 while we discussed about the replication. But those polymerases are not heat resistant, they are heat sensitive. For that reason only, especially for this PCR, we are going to use an enzyme called tag DNA polymerase, because this enzyme is heat resistant. Okay. So we are not going to incorporate this enzyme in between the reaction. So we are going to add all the reagents at a time, and that tube is to be kept into a PCR machine, and we are going to set a repeated cycles of denaturation, annealing and extension. So but when we apply a heat of 96 degrees centigrade, many of the enzymes will denature and lose their activity, but this is an enzyme which is heat resistant. It can withstand high temperature as well. For that reason only we prefer tag DNA polymerase when we go for a PCR. And extension or DNA synthesis. So this, what will this enzyme do? It will add DNTPs. Okay. One after the other the nucleotides will be added, and then the synthesis of DNA will take place. And this is carried out at a temperature of 72 degrees centigrade for 1 to 2 minutes. Okay. See about the beauty of this technique, where we can finish this procedure within a less time. But when we talk about the gene cloning method, it may take 2 to 3 days, and it is a time consuming process where it requires some expensive enzymes as well. But here in this PCR, we are going to synthesize the DNA in just 3 steps with a repeated cycles of denaturation, annealing and extension, where we maintain alternate temperatures, that is from 96 again coming back to 54 degrees centigrade, and again a slight rise in the temperature of 72 degrees centigrade. So this is all together form one cycle. In this way we are going to carry out 30 cycles, because in the first cycle we don't expect much of the DNA. Only the beauty of the method will be known after the repeated cycles only. That is, at the end of the 30 cycles we get billion copies of the DNA. As I already told you initially, that template DNA will be utilized to form a 2 DNA, then again 4, then again 8, then again 16, 32, 2 to the power of n. In this way at the end we get billion copies of DNA. These are the 3 important steps of a PCR. And this PCR can be applied in several fields, like in molecular diagnosis, in forensic science, and also in the evolutionary studies. So let's talk in detail about the PCR procedure. So the first step is that you need to take a small vials. We call them as PCR tubes. Okay, they will be very small, and they should be first sterilized by autoclaving, because all these reactions are to be carried out under sterile conditions. If not, if your DNA gets contaminated, you may not get the result, right? So for that reason you always need to maintain a sterile reagents. Okay, when we set up a reaction mixture, let's see what all is needed. First thing is we need to have a DNA template. Okay, so this is your DNA. Okay, you need to have a DNA template. Okay, this is your DNA template with this sequence. Okay, and you need to have a set of primers and reverse primer. Set of primers, that is forward primer and the reverse primer. I told you how to design a primer. You need to know the sequence information that is present in the template DNA. Okay, then you need to have a tag DNA polymerase. Okay, and then you need to have a water for setting up the reaction. Okay, then you can also incorporate tris HCl buffer, or simply we call it as a PCR buffer, and also some MgCl2, which can act as a cofactor to carry out the enzymatic activity. Okay, so we are ready with the PCR tubes, and we are going to add all these into a reaction tube. Okay, so we need only a less amount of the DNA, hardly we just add 1 microliter, and DNA template, just a minute amount of DNA is sufficient. Say for example in nanograms, like 10 nanograms is sufficient, and a set of primers around we add 10 microliters. Okay, and tag DNA polymerase, just 1 microliter is sufficient. So in this way we need to set up a reaction together. It will give you a volume of 50 microliters. So you are going to add all these reagents into a small PCR tube. You just vertex it, okay, to mix all the reagents. You will have a small vertex machine where you can keep the tube into that, and when you switch on the instrument, that will rotate those tubes. So just to have a uniform mixture of the reagents. Just 0.2 microliters is sufficient, but for the safe side we are going to use 1 microliter in excess. Why? Because when we apply a temperature of around 95 degrees centigrade in the step of denaturation, there is a chance that this enzyme may get denatured. Though it is stable at that temperature, but its half-life is just 30 minutes. Okay, but we are going to carry out 30 cycles. Of course, it will be completed within that time, but not to take the chances, we are going to add the enzyme in some excess amount. Okay, the rest, this is how we set up the reaction mixture. So then we are going to keep those tubes in a thermocycler. That is nothing but your PCR, and you need to set a temperatures at a time for both denaturation, annealing, and also DNA synthesis. So what temperatures you are going to apply? 95 degrees centigrade for 15 seconds, and for annealing 56 degrees centigrade for 30 seconds, and also for extension 72 degrees centigrade for 1 minute. This you are going to set prior to the start of the experiment. Okay, and you need to set that for 30 cycles, because no use of carrying out a single cycle. You can't expect much at the end of the first cycle. To get the sufficient amount of DNA, you need to carry out this repeated cycles for 30 times. Okay, remember, when you set a temperature for annealing, I told you to check with the melting temperature of a primer, and make sure that you shouldn't exceed the temperature of 65 degrees centigrade, which is not at all suitable for a primer to go and bind with the template DNA, right? It should be in between 45 to 56 degrees centigrade only. Okay, and here the temperature may vary from 95 to 98. That depends on the nature of DNA, as I already told you. And if it has more amount of GC content in your template DNA, you can extend the temperature to 98 degrees centigrade. And coming to the synthesis of the DNA, here actually the role of tag DNA polymerase will come into the action. And for that we are going to maintain a temperature of 72 degrees centigrade. Even at that high temperatures also, this enzyme can work efficiently, right? Why? Because this enzyme is isolated from an organism called Thermus aquaticus, which is a thermophile. You might be knowing about the microorganisms which are mesophiles and which are thermophiles. Phile means what? Love towards. That means thermo means what? High temperatures, like heat. We can say it as a heat. Thermo means heat, right? These organisms can grow at a high temperatures, and that is the reason we call them as thermophiles. And this organism, Thermus aquaticus, can live in hot springs. And so that is the reason the enzyme which is being isolated from this Thermus aquatic organism is being used for the PCR, because it can withstand the high temperature. So simply we call that enzyme as thermostable, because the organism is a thermophile which can resist high temperatures. Okay, along with the Thermus aquaticus, we can also use some other enzymes like Wnt polymerase and also PFU, which is isolated from Pyrococcus furosis, and also TTH, Thermus thermophilus. Again, these organisms are thermophiles which can resist high temperatures. So what's the role of DNA polymerase here? It is going to catalyze the synthesis of DNA by the addition of DNTPs, that is one nucleotide after the other to the growing polypeptide chain. So what is required here? DNTPs. Hope you are clear. So if you provide a DNA template, primer and tag DNA polymerase, can it carry out the process of DNA synthesis without the addition of DNTPs? No, right? Here you are also providing DNTPs, that is around 10 microliters of DNTPs. What are those DNTPs? DATP, DGTP, DCTP and DTTP. Okay, so this tag DNA polymerase is going to utilize these DNTPs, that is deoxynucleoside triphosphates, and based on the sequence that is present on the template strand, these DNTPs will be added one after the other. Okay, when you set up a reaction, you need to make a final volume of around 50 microliters. So make sure that you add all without any mistake. I'll just show you the diagrammatic representation for the better understanding. So this is the first step, that is denaturation, which is maintained at 95 degrees centigrade for 15 seconds. Okay, here what we are going to do in the first step, we are just going to apply a heat of 95 degrees centigrade for 15 seconds. And what happens? Strand separation. Okay, what happens in the first step after the denaturation? Both the strands of the DNA get separated. See, this is how both the strands get separated from each other. Okay, see, both the strands are complementary to each other and they are intact. Okay, they formed a duplex here. This is a DNA duplex here. So in the first step of PCR, that is in the first cycle, the denaturation takes place, nothing but the separation of the strand occurs, applying a temperature of 95 degrees centigrade for 15 seconds. And what is the second step? This is the first step. And coming to the second step, which we call it as annealing, that is nothing but the hybridization of the primer with the template DNA. Okay, here we are going to use two set of primers. Forward primer and reverse primer. Okay, in the second step, what happens? When we carry out the second step of annealing at a temperature of 56 degrees centigrade for 30 seconds, the primers will go and base pairs with the DNA where the complementary sequence is present at the flanking regions of the target gene. Okay, So this is the primer 1 and this is the primer 2. So these are to be designed in such a way we need to know the complementary sequence that is present on the template DNA. So here what is the requirement? We need to add a set of primers, that is the forward primer and the reverse primer, and make sure you add these primers in excess quantity to prevent the duplex formation. Why? Because here you are carrying out a denaturation step where you are going to separate the strands, but again you are reducing the temperature to 56 degrees centigrade where there is a chance of forming the duplex again. To avoid that problem, we need to provide these primers in excess, and these primers will immediately go and bind with the separated strands which are complementary to each other, forming a duplex here. Okay, hope you are clear. The primer will go and base pairs with the complementary sequence that is present on the template DNA, and it will form a duplex, preventing the formation of the DNA duplex again. Okay, coming to the third step. What is the third step here? Here strand has separated and here the annealing is done, that is the primer hybridization is done, and the final step is the extension or synthesis of DNA. So what is required here? DNTPs, that is DATP, DGTP, DCTP and DTTP, and along with the DNTPs we also require an enzyme called DNA polymerase. Okay, so what happens? Okay, observe carefully. In the third step, that is the extension and the synthesis of DNA, we are going to carry out this step at a temperature of 72 degrees centigrade for 1 to 2 minutes, providing all the DNTPs and tagged DNA polymerase. What this tagged DNA polymerase will do? After the hybridization with the primer, this tagged DNA polymerase, which is even resistant at this temperature of 72 degrees centigrade, utilizing all the DNTPs, it is going to add one after the other and catalyze the synthesis of DNA. This is the new strand of DNA, and here this is the new strand of the DNA. Okay, so what will be the information here? In place of T, the tagged DNA polymerase is going to add adenine, and in place of adenine here in the opposite strand it is going to add thymine here. So in that way, based on the information that is present in the template strand, the newly synthesized DNA will be formed, that is with the help of tagged DNA polymerase. This is how the 3 steps will take place in a PCR machine, that is denaturation, annealing and extension. So this is one cycle. Okay, what happens in the next cycle? Again this process repeats, denaturation, annealing and extension. Again in the third cycle this process repeats. In this way the cycles will repeat up to 30 times. So initially, so when you use a single DNA as a template, that is going to get doubled, 8, 16, 32 and so on. In this way the concentration of DNA gets doubled. So at the end of the 30 cycles, we are going to get billion copies of DNA. Okay. So for example, I will show you here. So this is the DNA, single strand of DNA. After the first cycle, it is going to get 2 daughter DNA. Okay. And after the next cycle, again, this will give rise to 2. Again, this will participate and that will give rise to 2. Okay, this way this process, the repeated cycles will occur. I will show you a graphical representation for better understanding of this procedure. If you plot a graph taking a time on the x-axis, the temperature on y-axis, so 0 degrees, 50 degrees centigrade and 100 degrees centigrade. So what happens in the first step is the denaturation, right? It will go up to 98 degrees centigrade for a certain time period, and then again the drop of the temperature to 50 to 56 degrees centigrade for a certain time period, and then again it will increase the temperature to 72 degrees centigrade. Okay, again what happens? This is the first cycle. What happens again in the second cycle? Again, the rise in the temperature to 98 degrees centigrade, and then again to 50 degrees centigrade, and then again to the 70 degrees centigrade, and this how it proceeds. This is the second cycle. So what is the first step? First step is the denaturation, which is carried out at a temperature of 98 degrees centigrade or 96 degrees centigrade. Then what is the second step? Annealing, that is carried out at a temperature of 50 to 56 degrees centigrade. What is the third step? DNA synthesis, carried out at a temperature of 72 degrees centigrade. This is after the first cycle, and again in the second cycle, again the repetition, denaturation, annealing and extension. This is how the synthesis of DNA takes place in a machine called PCR, that is polymerase chain reaction, and we can also call it as a thermocycler. Thermocycler in the sense we are using a different temperatures, starting with the high temperature and then reducing the temperature and then again increasing the temperature to 72 degrees centigrade. That means we are going to alternate the temperature here in this method. And when we talk about the functions of this DNA synthesis, in case of DNA replication, what happens? As we all know that the bacteria which are having a single ORI site, what happens? The process of DNA synthesis takes place at the ORI site, right? So to initiate the process, we need a machinery like helicases, ligases, okay, and we need single stranded DNA binding proteins, we need topoisomerases. All these are required to carry out replication in bacteria, that is in vivo conditions, but these are not required in case of PCR. Okay, in PCR, for the process of denaturation, we just apply heat. So when we apply heat at a temperature of 96 degrees centigrade, the double stranded DNA will be converted into a double stranded DNA will be converted into a single stranded DNA. But whereas when we talk about the DNA replication in prokaryotes, what happens at the first step? These helicases, single stranded DNA binding proteins, and the DNA topoisomerases will form a replication fork, that is they will make a duplex DNA by breaking the bond, hydrogen bonds. What the helicase will do? It will stretch the strands apart, right? Here we are separating the DNA duplex into a single strand by the application of the heat in case of PCR, which is done in vitro conditions. Okay, this is first important point. And coming to the second important point, we need primer. Here the RNA primer will initiate the process of replication. So in case of DNA replication in prokaryotes, RNA primer is required, right? But when coming to the synthesis of DNA with the help of PCR, here also we require a primer that is synthesized chemically. Okay, observe the difference. In both the conditions there is a requirement of primer. When it comes to the DNA replication, this primer is synthesized by an enzyme primase, right, which forms a complex with the single stranded DNA binding proteins, forming a primosome which will supply the RNA primers, right? But when it comes to the PCR, we are going to provide a primer that is synthesized chemically, that are going to initiate the synthesis of DNA. And coming to the other important point, when we talk about the DNA polymerases, here the DNA polymerase 3 will catalyze the synthesis of DNA. But when it comes to the PCR, we are going to provide an enzyme called tag DNA polymerase, which is a heat stable enzyme isolated from Thermus aquaticus. That is the major difference with the DNA replication and the PCR about the functioning of a PCR. In both the cases, the ultimate goal is the synthesis of DNA. But here, with the minimum requirements and in the in vitro conditions, we are going to synthesize the DNA with the help of an instrument called PCR. And this is carried out in vivo conditions. So remember one more important point here. you need to standardize the annealing temperature. Okay, for that you need to check the Tm value of a primer, and if you maintain a high annealing temperature, sometimes this primer will show a non-specific binding, and sometimes if you maintain a very less annealing temperature, you may not have a sufficient yield. So make sure you maintain a proper annealing temperature. Okay, now let's look into the difference between the PCR and the gene cloning. About the PCR and the gene cloning, I already told you in the beginning of the video about the gene cloning and how we perform the method of gene cloning. So I already told you that using this PCR technique, rapid cloning is possible. Here in PCR also we are going to make a multiple copies of the gene. So here we just need only less amount of DNA, that is in nanograms. But when it comes to the gene cloning method, it requires DNA in micrograms. And coming to the other important point, that is the PCR is carried out in vitro. Okay, that is in a test tube that is kept in a PCR instrument. But when it comes to the gene cloning, it can be performed both in vitro and in vivo, that is within the test tube or in a culture plate, and also it can be performed within a living organism. In vitro means outside the living organism, that could be in a test tube or in a petri plate. When it comes to the in vivo, especially when it is performed in the living organism, we call it as in vivo. Next important point is, it doesn't require any expensive enzymes, but the gene cloning require expensive enzymes. What all the enzymes required for gene cloning? We need restriction endonucleases, we need ligases, okay, which are to be again incorporated into a vector. We need a vector. So all those are the requirements in case of gene cloning. But when it comes to the PCR, we need only one enzyme, that is stacked DNA polymerase, and we doesn't need any expensive enzymes anymore. And coming to the other important point, this PCR can be performed in less time, less work and less skill required. What we need to do is just you set up a reaction in a test tube and you just put it in a machine. Rest the machine will work out on it. So it will carry out all the cycles and it will give you the product at the end. Once you are finished with the PCR, and if you want to identify the DNA product, what you are going to do? How will you identify the DNA? You need to perform a technique called gel electrophoresis. Agarose gel electrophoresis. What is meant by electrophoresis? Separation of compounds based on the size and the charge. Okay, when you perform this agarose gel electrophoresis, then you will get a band which can be visualized. Agarose will act like a sieve. Okay, when you have a DNA of different fragments and you want to separate those fragments based on the size and the charge, you are going to perform the technique called agarose gel electrophoresis. So the smaller molecules will move faster and the larger molecules will move at a slower rate, and then when you observe the gel, you will get a different DNA bands. In that way you are going to check your PCR product. For that reason you are going to run marker along with your DNA sample, and once you are done casting the gel and then running the gel, you are going to place that gel under UV visible trans illuminator, and then you are going to visualize the DNA bands using this electrophoresis technique, which I am going to discuss in my next video. When it comes to the gene cloning, it requires more efficiency, and this can be completed with a less time. That means denaturation, annealing and extension for 30 cycles. Hardly it may not take more than 1 hour. We can complete our experiment within a 1 hour time. But when it comes to the gene cloning, it may take 2 to 3 days, and moreover the error probability is less. Here we are designing the primers, and these primers will work efficiently, go and base pairs with the complementary sequence that is present on the DNA. So the chances of error are very less if you design a primer correctly. Here the chances of error are more. And coming to the other important point, it is a fully automated machine, but here all the steps of isolation of DNA and the insertion of the foreign gene into a plasmid, that is a vector, and then this vector is to be ligated with the DNA, forming a recombinant DNA molecule, and finally that recombinant DNA molecule is to be introduced into a host organism, which is a lengthy process and it requires time. That is to be done manually. So these are the differences. So you can make out that the PCR is best when compared to the gene cloning. Okay. Coming to the applications of PCR, this method of PCR can be applied in several fields. Like for example, it can be applied in molecular diagnosis, like if you want to identify any bacteria or any virus, we can apply this method of PCR. Now you know people were identifying the coronavirus with the help of the RT-PCR. So this technique of PCR can be applied in the field of molecular diagnosis, and it can also be applied in the field of forensic science. See, for example, in a crime scene, if you collect a blood sample, so though the blood sample is very less and the amount of DNA is in nanograms, I already told you the very small amount of DNA is sufficient to carry out this experiment, where at the end of 30 cycles we'll get a huge amount of the DNA, right? In forensic science, especially to identify the culprits, what forensic scientists do is they will collect the blood samples or a semen sample, or they will collect a hair sample which contains root, and from that root and blood samples we can also extract the DNA, because we all know DNA is present in each and every cell of an organism. So once we isolate the DNA, and that DNA is used to carry out the PCR, where we make a huge amount of DNA out of it. So then what we are going to do is, suppose there are 3 suspects, okay, and we want to identify who is the real culprit, and we are also going to collect the DNA from these 3 persons as well. Okay, now how many samples of DNA do we have? So from the crime scene we collected the blood sample and then we extracted the DNA. Along with this 3 DNA samples, now we have altogether 4 DNA samples. What we are going to do? So this sample is very less, that means the amount of DNA is very less here. We perform a PCR and then we increase the amount of the DNA. Now we have all the sufficient amount of 4 DNA samples. Now what you are going to do? You want to visualize the DNA by performing agarose gel electrophoresis. You will run a marker here. So based on the size and the charge, you will get different DNA fragments. For example, this is 100 base pairs, this is 200 base pairs, this is 300 base pairs, and this is 400 base pairs. And you have 4 more lanes here. And you are going to add DNA sample that is collected from the blood here. The blood that is collected from the crime scene. And you are going to add a DNA sample of suspect 1, 2 and 3. So all together you have loaded 4 DNA samples. So after you run a gel, you got one band here, one band here, one band here and one band here. So if you look into the picture of this gel, you got a DNA band of 300 base pairs. So this is the DNA that was isolated from the blood and the blood that is collected from the crime scene, which is of 300 base pairs. And the DNA from the suspect 1 is having a length of 400 base pairs, whereas the suspect 2 having a DNA, the DNA that is again collected from the blood sample with a length of 200 base pairs. And coming to the third person, observe carefully here, this band is matching with the DNA that is collected from the blood. In this way, this method is used to find the culprits. That is the application of PCR in the field of forensic science. So, and moreover, it can be also used in the evolutionary studies. Moreover, PCR can also be used to amplify some of the important proteins. like immunoglobulins and PCR, because it can be applied in several fields, like to solve the paternity issues and to identify the mutants. And whenever you perform a genetic engineering experiments, like you are going to transfer the gene into an organism, like you want to identify whether the gene transfer has occurred or not. In that case also you can use the technique of PCR. In that way you can apply this technique in several fields. Like these are some of the examples. This is all about the PCR in detail. Hope you understand this session, and I'll come up with the next session soon. And please take care of yourselves, and please follow all the precautions when you step out of the house. So now we all know that we are running through the tough times, so please be careful. Thank you.